So let's start. Let's start. Indirect Elisa. Indirect Elisa, which used to detect the antibodies, the blood of the person undergoing the assay for this test. For example, in case of HIV test, is allowed to clot and clear the serum with antibodies. We call that primary antibodies here. Simply, you have to remember, you have to recognize this as this is the test used to detect the antibodies. But nowadays, it's so easy to say. That means this is Corona time. That antibody tests are that all the way you are listening. So what they are saying that if I got some unknowingly, that means without symptoms, so many persons are affecting this virus, even they don't know whether they got it or not. That means that virus infected and he cured, but the person don't know that that surveys are saying like that. ICMR surveys and other surveys are saying like that nearly 2 crore people are already affected. How we can detect that? Okay, that how we can detect? We can that person is asymptomatic. How it is detected? By searching antibodies, thinking like so. If the virus is affected in the patient, if the virus is affected, then definitely he will have antibodies in them, in the person. If virus is affected. Okay, so that means we have to understand virus is infected to him, no symptoms in the person, but some fight is happened inside the body, isn't it? Some fight may happen. That's why he is able to produce the antibodies, he recovered. That means here he is not even affected. Some thousand entered in the body and he produced 1,000 antibodies and more than 1,000 antibodies he fighted and he won. But he don't know he won the match. He's smiling because that he is infected, but he don't know he is infected because no symptoms exposed. That means he is able to kill how many viruses are entered inside before they multiply. Even they are multiplying, he is able to kill. So what you can detect in him? Antibodies. If we find antibodies, we can say he may get infected. So that's the story behind this. Use it to detect antibodies. Okay. So for that, what we need to have in the laboratory? We need to have in the laboratory, we need to have antigens. Okay. So now let me draw that same microtitry plate, that antigens. Okay, so let me draw the antigens. That means when we are testing for HIV virus, the HIV virus antigen particles you should have in the laboratory. So that I am showing here. HIV virus antigen particles. Okay, so this is antigen which I am showing here, which now who attached to the microtitry plate, I am going to detect antibodies. That's why I made antigens to attach. Okay, so I made antigens to attach to the well, microtitry plate well. I attached antigens. Okay, so what we have to do? We need to add the patient sample. A known antigen, I made it here. In case this is HIV case, I'm doing actually. The HIV antigen in this case is added to the well. So we use a word called adsorb. Adsorb. That means antigens adsorb to the well. That means attached to the well. HIV antigens adsorb to the well. Microtitry plate well, they adsorb. Okay. So think we can increase the font. Yeah. Adsorb to the well. Next, second one. Second one, what we have to do, we need to add the patient sample. Patient sample for detecting what? For the detecting the antibodies. If he is infected, he may develop antibodies, isn't it or not? So that's why patient's anti-serum. Patient's anti-serum. Serum which contain the specific antibodies. Anti-serum means that which contain specific antibodies. Again is that particular antigen. Specific antibodies. Patient's anti-serum is added. That means if it is if he is infected already, that what happens that anti-serum containing antibodies will attach to that particular, the primary antibodies or complementary antibodies which are there, they go and bind with them. So I am showing, should change color. Yeah. So that the patient's anti-serum which contains specific antibodies, if present, if antibodies present, a B, small b we have to use. If antibodies are present in the patient sample, they attach. Present, they attach to the antigen of microtitry plate. They attach to the antigen of microtitry plate. So what we have to do next? Now, what we did actually, the antigens are there which attach to the well. Patient sample is added. Then the antibodies which already we have to say that as primary antibodies here. Now, enzyme linked antibodies we need to add. Enzyme linked anti-human immune serum. That means which can bind with the antibody. So what do you say that as secondary antibody? Okay, that antibodies are called secondary antibody. So I am going to add the second one. Enzyme linked anti-human serum, which means which contains secondary antibody. Enzyme linked secondary antibodies, which means which can bind with that antibody, are added. Okay, so let me show you that first of all the primary antibody which is there in the patient, and then after the secondary antibody which is enzyme linked. So is there and they go and bind with the primary antibody. So that's it. So which I mean finally we have an wait I have to change color. Finally patient antibody, primary antibody is there, and next enzyme linked antibody is also there. So for that you add substrate. When we add substrate, the reaction the which that the substrate binds. So let me show you here that add substrate. This is very important. Then the final result. Final result. Fourth one. Add substrate. If it changes the color, add substrate. If color is changed, what you can say? Color is changed, which means positive. Again, the scenario get changed. If color is changed, it is positive. That I am going to declare it now that this is positive test. Positive test. Okay, so next, let's do the negative test. There you can understand the scenario. Let's do the negative test. The same, that indirect Elisa, the same old, that negative test I have to do. 4 samples microtitry plates I am taking, and as usual that antigen we are making attached to the well, antigen is attached to the well microtitry plate. Then the patient sample is added. When the patient sample is added, so already first of all let me draw all antigens on the plate. The antigen attached to the microtitry plate. So second one, what we did, that patient sample is added. If there is no antibody in the patient sample, no one will bind. Then after, when enzyme linked secondary antibodies are added, when enzyme linked secondary antibodies are added, they can't bind with this antigen, isn't it? So that means they have to bind with primary antibody. So when we absorb the free fluid, that will go off. When we add substrate, there is no change in color. Okay, this is called negative test. This test we say it as negative test. So let us describe the negative test. Number one, that patient sample. No, in laboratory, that antigens, specific antigen, that means HIV antigens, HIV antigens attached to the well. HIV antigens attached to the well, microtitary plate. Number two, patient sample is added. Patient sample, what we are detecting in patient sample, serum containing antibodies. Patient sample is added. If 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 we have to say, they should not say directly that there is no. If there is no antibodies, if there is no antibodies, they won't bind, isn't it? If they are present, they will bind. If there is no antibody, they won't bind. Okay. Next, number three, the enzyme linked secondary antibody, and that secondary antibody means which can bind with antibody, is added. Okay, is added. So what we have to write, if it is added, if there is no any, if there is no primary antibody, patient's primary antibody, they can't bind. If they are not present, they can't bind. That's so simple. Okay, next, the fourth one, add sample, add sample, add substrate. When we add substrate, no change in the color, no change in the color. That means we say that is negative. That means we say that is negative. Okay, so similarly like the previous one, here also I want to show you that both positive and negative in one place, both positive and negative in one place with the bigger diagrams. So antigens are attached to the well. We have HIV antigens in the laboratory, that to search the HIV antibodies in the patient. So if patient having that antibodies, if the patient is having that antibodies, if the patient is having that antibodies, they go and bind with that antigen. Okay, so then next what we have to say, enzyme linked antibody is added. Enzyme linked antibody is added. So that enzyme linked antibody will, secondary antibody will bind with the primary antibody. Okay. So in these conditions, if we add substrate, the substrate, as I said to you in the earlier classes, which can bind with the enzyme, we say substrate, beta galactosidase, peroxidase, whatever, that enzyme that which can bind with that we can say substrate. So if there is a change in the color, when you add substrate, the substrate will go and bind with that, react with this antigen containing and that coated enzyme on the above on the antibody, the secondary antibody who is coated with that enzyme, they react and that makes a change in the color. Then in this case, in this particular condition, if there is no that patient antibodies are not there, they can't bind. If they can't bind, enzyme linked antibodies are also can't bind and can stay in the microtitary plate. When we absorb them, the free fluid will be that absorbed over. So add substrate, add substrate, then no change in the color, no change in the color. If there is no change in the color, So what we can understand, if there is no change in the color, we can understand that is negative. So the first one is positive test and the second one is negative test. So finally, we are the completed that indirect ELISA, with this particular. So then this is all about the tests of positive and negative direct ELISA and indirect ELISA.